So this is the first webinar the NIU, Nnamdi Azikiwe University, Genomics and the Bioinformatics Consortium is hosting. And hopefully every month we should have a webinar from the foundation of bioinformatics, and we build it up from there. Of course, the last will be in September, and hopefully we believe you will continue to join as we start from the basics. So today we are starting with the applications and the essentials of bioinformatics. This is a start, and we build up from there. Very important for many of us that are new and those who are still considering running a career path in bioinformatics. So happy to have you today. And here we have Dr. Akinola, okay, who is going to present today. Akinola A, Alafiatayo. Yeah, so Dr. Akinola had his BSc in biochemistry from Ahmadu Bello University, Zaria, and accelerated quickly to MSc in genetic engineering and molecular biology. Yeah, then in PhD also genetic engineering and molecular biology at University Putra, Malaysia. And since then, he's been working on transcriptomic analysis of premature skin aging. So currently, he's a postdoc fellow at Discipline of Genetics, Faculty of Medicine, Memorial University of Newfoundland, Canada. And that is where he's going to lead us today in this discussion. So currently, his research is on molecular genetic analysis of autism with focus on structural variants, identification, and gut microbiome study using the next generation sequencing technique. So, and we hope that in the course of the year, he will also be our resource person in the NGS techniques. I think I've mentioned that to him, but we have not discussed extensively. So, Dr. Akinola, you are welcome to share your screen and start the meeting in full. So currently we have 15 persons, but this is tied to time and we have to go ahead. Yeah.
Okay, all right. Thank you, Dr.
So maybe
Moses.
Aniedu, you could stop sharing so that, yeah, so you could share now.
Okay.
Yeah, you could go ahead now to share your screen and move on from there.
Share. Have you stopped sharing?
Yeah, we have stopped sharing for you to share.
Okay. Please kindly confirm again if you stop sharing, because I cannot still sharing.
I have stopped sharing, then I've also enabled you now to share.
Okay.
Yeah.
Okay, all right. Can everybody see my screen?
Yeah, but you can make a full, yeah, thank you, perfect. Yeah.
All right, I will say good morning from this side of St. John's, Canada. Good afternoon to Nigerians. I've been introduced. Actually, my name is Akinola. I'm presently a postdoc at the Memorial University of Newfoundland, where I'm actually working in the Faculty of Medicine, and my research focus on structural variants, which is mostly the use of next generation sequencing tools and tech in identifying structural variants in autistic kids, and also in collaboration with studying gut microbiome for personalized medicine or personalized care. So to go straight to the program, to the event of the day, I've been given a topic to speak on applications and essentials of bioinformatics. So first of all, I will say that by the end of this webinar, I would think every one of us should be able to have some one or two things to learn about the field of bioinformatics, and that is to understand bioinformatics as an interdisciplinary field, then to understand the essentials of bioinformatics and the possible applications of bioinformatics in our daily life. So if we were to start, let's start with the genome. And I believe that every one of us, or everybody, or if not everyone, who have background, we must have one or two background in contact with what we call the genome, which is called the DNA, which serves as a manual of life. Every living system actually have what we call the genome. And in human, this genome is actually divided, composed of 23 pairs of chromosome, which encodes for virtually all the proteins in the body system. All right, and these proteins are being coded for by 4 basic nucleotides, which are basically adenine, thymine, cytosines, and guanine. Now, the total human complete genome is estimated to be about 3.2 billion bases, which are made up of ATG. Then, out of these 3.2 billion, only 2.9% of the total genome encodes for proteins. Now, if you see from this diagram, usually from a cell, from every cell, we actually we have the chromosome in there. Then the chromosome composes of the nucleotide, which are actually linked together in pairs. Then, which following the central dogma principle are actually translated. They are transcribed, they are translated into protein, and the molecular machinery of the body. So the foundation, the basis of everything we're going to talk about, mostly in bioinformatics, is actually going to be based on this principle, on this life machinery. Now, as we all know, as a living system, or which comprises of bacteria, archaea, and eukaryotes, we are all living systems, and every one of us are made up of genomes. And as I've explained earlier, genomes are actually a series of nucleotide sequences joined together. So bacteria, eukaryote, plant, fungi, we are all composed of these 3 or 4 nucleotides. Then, recently, 2 events actually occur that actually change the face of the profession of bioinformatics. And one of them is the complete of the Human Genome Project, which was completed in 2003, where the complete genome of human was actually sequenced and completed. It was actually initiated by the US, by the Department of Energy and NIH in a joint consortium with 5 countries, which are US, UK, France, Germany, Japan, and China. And the total budget was supposed to be 3 billion in total to sequence a single human genome. But that was 2003. But now, I think you can get a complete genome sequence as low as $1,000. You can do that now at the moment. So that was one of the major breakthrough in life sciences that actually put forward or bring to limelight the field of bioinformatics. Then, secondly, I think just of recently, that was 2013, if I'm not mistaken, or 2013 thereabout. Or no, I think it should be 2005. The advent of the next generation sequencing techniques also came into being. And then this is one of the science tools or innovations that actually revolutionize the life sciences, in which you can sequence a complete genome of an organism in a shorter period of time and with minimum amount of cash, of money. So these two events actually was what actually bring what we call the field of bioinformatics. Although bioinformatics has been existing or has been around as far back as the 1970. I think it was just the word bioinformatics was first used in 1970, where I think the genome of a yeast was sequenced. So, but it actually came to importance when these two events occurred. So now that boils out and takes us to then what is bioinformatics? Now, from what so far, bioinformatics can be actually defined as the union between computer science and molecular biology. It's actually a field, an interdisciplinary field that develops and applies computers and computational technologies to study biological questions. For instance, you have some tools, because the genome is totally large. It's not something we can just do. Even we can do ordinarily with a mere just going through the bases one after the other. If you have to do that, I think in 50 years, we've not completed the whole genome. So we needed like a tool or a machine that could just help us as a life science, as a scientist, that could scan through the whole genome and look for special uniqueness in those nucleotide sequence, which are the basic unit of life. Now, to do that, as a life sciences or as a biologist, we don't have the knowledge or the capacity or the skills of computer science. Then the computer science really don't have the skills or the knowledge of biology. So what happens? So both of them need to now match together, or they need to form like a point where both of them can actually work and come together to obtain or have a particular set of goals. Now, that's the first about interdisciplinary approach. Then it's also, you can also be said to be like the information and data management of the biological sciences. For example, for instance, the big data, it was going on all over now. But when you have your data, how do you manage your data? You can't just have a data and to just throw it out there. Data needs to be managed, data needs to be stored, and then information should be able to be retrieved from the database as quick as possible. So what happened is, as a bioinformatician build tools to actually store this data. That's where like a warehouse where this data are being stored. We call that a database. And then those tools are also being applied in the analysis of biological or answering biological questions. Am I still being audible? Are you guys?
Yeah.
Okay.
Go ahead.
All right. Okay, because I can't see myself. So now the field of science in which biology and computer and information technology merge together to form a single discipline is what we refer to as maybe bioinformatics. For example, as a biologist, you collect data. You have your DNA, you have your protein sequences, you have gene expression data, but you are faced with the problem of how do you get meaningful information from these sequences that is made up of ACTG in billions? How do you make reasonable, make sense out of it? That's number one question for a biologist. Number two, you have the data. The data cannot actually be stored in a single laptop. You need a big data storage capacity. Then how do you prepare? How do you make the database where you store this data for future use? How do you get that? So that is being lacking in the field, in the aspect of the biologist. Now, if you come down to the computer science, yes, you have the computational skills, you have the statistical skills, you have the software, you can develop the algorithms. But what about the data? Can you give meaningful interpretation to the data? So these two fields comes together to form a biological question on how they could just analyze molecular data. Sorry, I think I'm done with it. So now in the bioinformatics, as I've said earlier, from the genotype, which is from the gene, down to the phenotype. Okay, DNA from the genome is being transcribed into RNA, protein, and from there we have the molecular network. So this, from the DNA genome, from the DNA of the genome to protein, which is we call the basic central dogma principle of molecular biology. When you have the data, you have your DNA, you have your sequence. You need to align your sequence to a data or to map. You want to determine the motif of your gene. Then you want to check for similarities. So these are areas in which where the biology part of the bioinformatics comes in. Then you have the gene, finding of gene, finding DNA methylations, we have evolutions. Then you have a place where you have a differential express gene where you study the total transcript or RNA in a particular system or at a particular treatment. Then if you move ahead, you saw how where you study the protein-protein interactions, where you actually look at how do protein interact with themselves. What are the transcriptional binding factors that actually regulate the expression of these proteins? Then some other metabolic pathways and signaling pathways. Then we can go ahead to form what we call the population genetics and the human genetics, where you actually look at hereditary, how some mismatch or some mutations in the genome or in the gene actually affect the phenotypic appearance or have effect in an individual. Then we can also simulate cells. So this is where the bioinformatics comes in. Then when you look at it in the informatics part, which is in the bio, these are the data management, where you need to manage the data from your research, where you need to actually get your data, transfer your data into the databases, then for future use, and then we have the data computation, where you make use of algorithms or you build algorithms, you build software to actually solve some of those problems that I think that's been lacking. Then data mining, how do you mine those data from the database to get a meaningful biological information that will be useful to maybe to individual or to the organisms. Then you can actually move ahead when we have the modeling and the simulations. This is mostly predictive models in which you simulate a biological environment or a microenvironment of an organism. So now we can now say that bioinformatics is a multidisciplinary field that comprises fields like biology, biochemistry, chemistry, computer science, engineering, mathematics, and mostly statistics. And all these fields, they come together to form the field of bioinformatics. But bioinformatics is not really the same as computational biology. So there's a kind of confusion. People think maybe bioinformatics is computational biology. Bioinformatics actually deals with sequences using computational approach to deal with sequences that happens to be like a flat file, whereas computational deals with all the computations of biological sciences. For example, maybe there's what we call geomapping. All those ones, they are not bioinformatics. In other aspects where you don't actually deal with molecular biology, because bioinformatics, another name could be molecular biology informatics. So now we go down to the applications. This field has been in existence and is currently being used and is a thriving field. Now, where and how can we see the application of this field? For example, okay, for instance, biocuration, which is what I've actually mentioned before. How do we curate your data? When you have a data, you generate a huge data, then you need to curate it. That's you need to store and individualize. So bioinformatics help us to store. Then from there, we can actually retrieve those data. We can analyze, interpret, and also integrate them into biological information. When we get that, from there, we can actually store them in the data repositories. Now, how do we, once we have those data, and we actually want to store them, there's what we call the database. The database is actually from the genome. We have the gene predictions, and then which is actually when we sequence and we map this back to the genome, where we will receive an information. This information will be in form of sequence. Then this sequence can actually be stored in the database. And some common databases we have is we have the popular the NCBI, which store virtually nucleotide sequence. Then we have the Swiss Pro, which basically deals with protein. Then we have the KEG for pathways, mostly for metabolic pathways, and we have the Motif database, which actually identify motif. That's the interaction of the protein binding site on the DNA. And then we have the PDB, which is a protein database, where the protein that their structural has been elucidated. We have their structure, we know them. So these, we also have a particular database for them. And we have the UCSC, which is basically mostly for nucleotide sequence as well. So we have thousands currently. We have currently we have thousands of databases for different organisms. We have the Arabidopsis, we have for plants. So it just depends on the kind of research you are doing that would identify the kind of database you actually be utilizing. And then you need the basic tools or skills of computational biology or computer science for you to be able to walk through these databases easily without stress. And then one thing again about the database, you should be able to deposit data and be able to retrieve data from these databases. So the database are actually classified. We have the primary database, and then we have the secondary database. The primary ones are actually the gene banks, which are the consortium of the gene bank, which is from the US, the NBL, which is European Bioinformatics Institute, and the DDB, which this is Japan's. So these three consortium, they work hand in hand in a regulating entry of data into NCBI, because they both regulate NCBI. Now, if you drop a sequence into DDBJ, it's going to be linked to the gene bank, and then so once you go to any of these database, you are sure of having the same information because they are both, the three of them are linked together. Then the PDB are databases where you actually get protein structures for your molecular docking. I think that's actually common in Nigeria for your drug or whatever protein binding site. This PDB, you have a wealth of structural some proteins, or actually, I don't know how many proteins, sorry, that are being deposited, but you basically have the major proteins here in which you can form, you can determine compounds that can bind to some active site. Then we have the Medlin. Medlins are basically the literatures where you can get literatures. And then IMEX for protein interactions, how protein interactions actually work to and their effect. Then when you look at the secondary curated database, this is curated. What they mean by curated, it means that you can actually extract information from them. This is for archive, where you store. This is maybe you want to retrieve information. For example, the RefSeq. The RefSeq means reference sequence, which are assumed to be like a standard sequence. If you want to, for example, you have a sequence, you want to see which part of the genome is this sequence from, you can use the reference sequence to do that. And then you have the UniPro. UniPro is just like a Swiss protein, as I've said earlier, for protein sequence. And then we also have the taxonomy database, which mostly you can use to store your sequence from any organism, and then you generate your final genetic trees from there. Then the ProSite for binding. And then I think we can just hide the listing continue and continue. So it depends on your area of research. Okay, yeah, this is an example. These are just an example of created and review annotations where you can actually get, so these are some of the information you can get on the database. For example, we have the functional, you can get the function of protein, the subcellular localization, where this protein are localized, their post-translational modifications. Maybe you have a particular protein you're working on, you want to see after the expression of this protein, what happened at the post-translational level. Then you can get the domain sites from all of those, any protein you're working with. Then with the secondary structure, the quaternary structures, all these can also be obtained from the database. And then we can actually also associate disease-causing genes or disease-causing proteins, disease-causing variant, or a wealth of information we can actually retrieve from there. So, okay, this is PDB. PDB is just basically a database of protein structural proteins, where the proteins are in 3Ds, where you can actually you can dock a compound to see to check the affinity of those compounds. Is mostly can be used for drug discovery purposes. And the recent application since after the advent of the next generation sequencing technology, then the field of bioinformatics has actually boomed or flourished. And now I want to go into where does NGS come in bioinformatics. So one of the first place or the initiation is we'll talk about the genome informatics. This has to do, this is like a cardinal or the main point of NGS, because you have a cell actually they have what we call the chromosome inside the cell. These cells are made of DNA are made of nucleotide. Then we go ahead to sequence. So basically, any research that has to do with us manipulating the DNA, checking the sequence, knowing the aberration in the sequence, all these are all classified under what we call the genome informatics, where you try to study and retrieve an information from the genome. So the genome is just like a book that contains 3 billion base pair, which is how the manual that give instructions to the kind of proteins that will be expressed, or how which protein will function. Now, if there's any aberrant in the way the sequence are arranged, if there's what we call the switch or mutation in one of the sequences that actually affect a particular protein in a protein coding region, or maybe the exon, this might actually affect an individual. Maybe it could cause sickness, it could lead to any other thing. So now the function now is, or the onus lies on us to find, okay, out of these 3 billion bases, how do we know exactly this spot where this thing this mutation or aberrant is actually occurring? So that is where the machine comes in, which is the computer, because we can, as I've said earlier, we can scan through, and we will need what we call a reference sequence in which someone will believe that this person is accepted or is agreed to have moderately or better sequence where those where we can actually identify the missing link. So in the database, we have a reference, we have a standard reference sequence. I think the current one is human genome sequence 38, which has been there since 2013. So the question is, we have this. So what else do we do? After we do our next generation sequencing, we receive our sequence in this format. It's going to be in a FASTA file, which is going to be thousands or millions of reads and reads and reads. So how do we now put this together to make a meaningful information? That is where the bioinformatician comes in. So one of the first application of where we apply bioinformatics is where we study the whole genome of an organism, which is we try to determine the total sequence, DNA sequence that comprises of that organisms. These techniques was actually introduced into the lab or into clinical purpose in 2014, where it was used in the US. But before then, it was just used for research purpose. So as time goes on and they begin to perfect it, and they say, okay, this could have a better application. So it finally end up finding its place on the bedside. So now with this, we can actually detect mutations, and some of the mutations, that's mutation frequency, and some of these mutations, some of them might be lethal, some of them might just be ordinary mutations. Then we can use to detect what we call rare genetic disorders, because there are some disease that actually occur as a result of random mutation, which we don't really have any record, or people who have those kind of sickness are rare. Maybe we could just have like 1% or 2% of the population. So those kind of sicknesses are very, very rare to diagnose. But with the help of whole genome sequencing, we could actually diagnose those kind of conditions. From that, we could profile solutions. And then we could also, it's also a good avenue for us to detect what we call the rare variant association study, where we actually detect some variants which are also disease-causing variants, which are very, very rare, which which are not something we know. So these are tools basically that can be used, that can be harnessed by the clinical communities, which is with the help of a bioinformatician. And a medical doctor do not have this kind of knowledge or this kind of information at hand. So they would still need the knowledge of bioinformatics. But, things move, things change also. So in the kind of an advanced country, they've started incorporating the study of bioinformatics into medical professions, which they actually do now here. So because of the advantages of this we have. Then the whole genome also is being used in what we call genome-wide associational study, where we actually look at variant or mutations in a group of people or in a community to actually identify if you can say it's called a mutation, because there are some mutations that they are unique to everybody. So we want to identify this mutation to serve maybe like a marker. For example, let's say we have about, let's say for instance, we have about 500 people having a particular sickness, and all of them are having a particular variation in their variant. So this can actually be used as a tool to identify that mutation or that variant, which could end up becoming a biomarker for diagnostic purpose. And this is going to be done also with the help of the machine. We can't just sit down. So this is how it works. Maybe you have a kind of patients, patient 1, 2, 3, with their sequence. And we have the control, which are people who are unaffected by that sickness or whatever, or by that disease. So by the time you align their genome together in this way, and you also align, you try to find out what is unique to the people that have the disease, but that is not present in the control sample or the control patient. Now, if we look at, in this case, there is a mutation here in which we can see virtually people that are carrying a particular disease, their adenine is being replaced by thymine. Okay, so this is actually a good kind of that we can conclude, but we don't just conclude based on in silico approach. Okay, so once we have the result like this, we still need to go back to the wet lab and validate if these are really true, because we can't rely on the machine or the computer 100%. But the computer, what they do, they aid us, they give us, like, they beam a light into what we are doing to make our research faster, to make it more economical, and also to give us a direction. So another application where bioinformatics also is applicable is in the area of RNA-seq, which is RNA sequencing. RNA sequencing is when you actually extract or get the whole total RNA. It could be mRNA, transfer RNA, all the mRNA in a system or at a particular treatment have been extracted. Then you sequence them to compare the transcript at a particular maybe treatment or in a particular sickness that is actually affecting an individual. Or this is also a place where you can actually use to study plants. Also, this is also applicable to plants. Then what happened is, how does it work? How it works is you get, let's say, okay, let me use an example of plants. You have a particular plant that thrives well in less rain or in a drought environment, which is abiotic factors. Now, okay, let's use that as a pest. Plant is being affected by a pest. Then we all know that once a plant is being affected by a pest, the body system will actually try to defend itself. So when the body system tries to defend itself, or the plant system, then at that period of time, some genes will be switched on. So that particular time that those genes, or those that the plant is going through, that is like a gene of survival. So you could harness those mRNA at that particular time, sequence them, then know what kind of gene are they. So these could actually aid us in designing a disease or pest-resistant plant. And also in the area of when we talk about disease alterations. Let's say A has a particular disease, let's say cancer. You get the tissue from the cancer, then you sequence it. When you sequence it, you compare to unknown, you compare to a primary cell, a cell that is not cancerous, to check the difference in their expression level. It's actually called gene expression profile, to check their expression level. So that gives us an insight. Then once we do that, we can move ahead to study the protein network. How do those mRNA actually come together? Those proteins that's been transcribed and translated, how do they function together in a complete network? But currently, the popularity of this is becoming less, and it's been taken over by what we call the single cell RNA, due to problem of heterogeneity of this sample. Because what happened is in a single cell RNA-seq, the problem of heterogeneity has actually been solved in such a way that individual cell in a particular tissue is being segregated and then sequenced individually. So, for example, if you look at the blood cells are made up. The red blood cell is made up of some other component of cells inside the white blood cells and the red blood cells and everything. So what happens is when you get those things, for example, if you take a tissue, let's say you take a tumor tissue, if you take a tumor tissue from the brain or from the lung, all these are made up of different cells. So these tissues will be segregated into their particular cells. Because why? Because in the previous RNA sequence, we cannot really pinpoint which of the cell is actually being affected, because each individual cell is an entity on its own. So these techniques went ahead to see how they can actually segregate these cells and know which of the cell is being affected, which of the cell is growing apparently in what they call it, in a pathological conditions. So, and that give us, we have a list of genes, and then we can use principal component analysis, which is statistical aspect, to group these cells together and study the effect of each of them. That is, and all this also has been done, is being done by the computational approach. Then epigenetics is another area of NGS where bioinformatics comes in. As we all know, these are changes on the gene expression profile that does not affect the sequence of the DNA, but actually affect the expression of a protein. And these epigenetics have been attributed to environmental causes. Maybe it could be smoking, lifestyle, how do we live, or exposure to some other chemicals, or naturally, it could also be, and it has been known to be able to be transferred from generation to generations. So, and with this, the epigenetics, we have the DNA methylations, we have the histone modifications, and then with other acetylations. This helps us to actually control how a gene is being downregulated or upregulated. Let's say, for instance, let me take an example of also a cancer. When you look at the epigenetic profile of a cancer cells, you find in a particular region, we call it CPG islands. In this CPG island, you can see maybe it's actually at the upstream of the gene. And this CPG island, you can see that they are high, they are all methylated. So in that situation, that methylation means that the transition factors cannot bind for the expression of that gene. So what happens is, if that particular gene is actually a tumor suppressor gene and it's not being expressed, we should know definitely that that person is prone to cancer, or is probably going through, or in the future he might have cancer. So in this, we can actually use it and also to detect the methylation profile to see how gene expression or causes a disease, or solve use in some areas of plant also. Then also, this is also is one causing what we call S chromosome inactivation, because we have pair of chromosome, and the two chromosomes cannot actually be expressing at the same time. So one of the chromosome is totally methylated, so that information from that particular chromosomes would not be expressed, whereas the other one, the other chromosomes, then that's when we see the phenotypic appearance of that. So this basically helps us also in diagnosis and also some treatment of cancer. But this also cannot be solved without the issue of without the aid of a bioinformatician or the use of the computational biology or a computer, because it has to go to the DNA level, in which you sequence the DNA, you try to identify the regions of the DNA that are actually methylated. And this is in the, you do this in the whole 3.2 billion bases that you have in the genome. Then another aspect is the metagenomics. Metagenomics is just a study of genetic materials that has been retrieved at a particular point in a particular environment. For example, it could be environmental. Maybe you have oil spill or whatever, or it could be like the gut microbiome, where you can sequence all the microbiome presence in your gut through the saliva, or it could be from the feces. So it depends. So you are trying to actually study the genetic material that is actually found, or the genetic material of all microorganisms that are found in a particular environment. That's the concept of this. But the thing why this is actually advantageous is because majority of the microbiome that lives in the gut are not culturable. So now, if you don't culture them, or by the time we culture them, we add some chemical, we really don't get the true presence or the microbial diversity of that environment anymore. So what happens is, what we do is to just straight away we extract the DNA or the genetic material. It could be DNA, it could be DNA, it could be RNA. From that, all microorganisms in that environment. And what we do after that, we take them back. Once we have the genetic material sequence, then we'll take them back to the lab or to your computer, and you try to identify the presence of this species in what proportion in that particular environment. And that gives us information about the metabolic pathways, the metabolite that is being produced by those microorganisms. So we can classify them either from the phyla level to the level of species, just basically using their genetic material without culturing. So that is one of the advantages. So those unculturable microorganisms can actually be studied without issues. And this is in real time, because if you need to collect your sample, you move them to the lab, you start culturing, isolate. By that time, a lot of things must have gone on. So you might not really get the true picture of some of the species that are present in that environment, which might give us, at the end of the day, a false positive result. So one of the major area where it's being used in the Human Microbiome Project, where they try to actually study the microbiome from the present from unborn child to an elderly person, because at each particular time, the microbiome we have or we carry tells much about us. Because there's even this saying that they say that there's this report that said that the total microbiome we have in our system is even more than the whole cell, times 10 of the whole cells we have in our body. So with the microbiome, we could do a lot. And one of the major places where this has been used is what we call the fecal microbial transplant, which is a new area that is being tried, in which, for example, people who have been Clostridium difficile are people who have been on antimicrobial for so long. So they've actually the antibiotics has actually affected the diversity of their gut microbiome. So what happened in that case is that they have given opportunities to other pathogenic microorganisms to grow, so which affect them. So for us to correct that, we need to balance the human gut microbiome ecosystem. So you can get a fecal from somebody that is healthy, which the fecal has been screened, then to replace or to replenish the gut microbiome in the rectum. And they've found this, and they see that within 2-3 days, people actually picked up, and they end up getting well or getting healed. So so this is also being used. This is also being used in bioinformatics, in health wise, and the skills of bioinformatics or the application of bioinformatics is also being applied here. So it was also in the area of in veterinary studies, they've been able to use the microbiome study to identify about 27,000 new genes from cow, in which they can degrade polysaccharide, which is becoming a good application in biofuel, where we can actually degrade polysaccharide to form energy, to produce energy or to produce power. Sorry, Dr. Moses, how many minutes do I have left? So say I can just rush through. You still have more time?
Yeah, just 5 minutes so that, we'll take
Okay, so I need to, because I have more slides. So in the area of our crop applications, it has also been used in the way you compare comparative genomics of plants, where you compare the genome of a particular plant. For example, we have the model plant. The popular one is called Arabidopsis, which because bioinformatics are able to tell us that these plants' genomics are conserved. So information from the model plant can be actually applied to another plant. Then in this area, we've seen it has been used to improve the quality of vitamin A fortified rice, where we can identify the gene bioinformatically, then we can actually infuse it into a tomato or vitamin A to produce vitamin A, or to extend a shelf life. Also to produce a drought-resistant varieties that's to a biotic or biotic stress. Then in the area of drug development, computational tools have been used to identify and also to validate new drug targets, which a medicine can be specifically targeted to that what do you call it, that point, oh, I don't know how to put it now. To target those, the drug can be used to target the time I want to use the protein active site of that particular, maybe if it's what do you call it, if it's a parasite, if it's a epidemiological, that's to do with virus or disease or any parasite. So you can actually look for a drug that can actually target those proteins.
The essential proteins in the parasites, yeah.
Exactly. So, and then personalized medicine, where we can use bioinformatics have been used to identify how each individual genome responds to what we call medicine. So, because we all carry different genetic variation, and we respond to drug differently. So, using in the field of pharmacogenomics, we can use a bioinformatics to actually analyze your genome and see how you can actually respond to drug and at what dosage. And then you can prescribe from the onset of a disease, you can actually prescribe the correct dose and the correct medicine to solve that issue or problem. Then, so in conclusion, so I can conclude that bioinformatics will guide and help every molecular biologist and clinical researchers to capitalize on the advantage brought by this computational biology. So we need to hone our skills in the area of this, equip ourselves with skills and tools available to us as a molecular biologist. And in the area of clinical research, it will be easier to easily identify or diagnose a disease, and then it will help us moving from the laboratory bench to clinical practice with the use of bioinformatics tools and skills. That, I would say thank you to everyone who is here. and then the floor is open for
questions. Yeah, thank you, Dr. Akinola. Thanks for, yeah, so there are many applications of bioinformatics and the essentials of it that probably it wasn't my thought earlier before now, but as the presentation goes on, it became clear to me, even though one may not be able to apply in all the areas that have been mentioned, but you may need to get focus on one research area like metagenomics or epigenetics or as the drug development or as the case may be. Yeah, maybe you could stop sharing your slides, Akinola.
Okay.
Yeah.
So, sorry about that.
Yeah. Go to new share. Yeah.
Okay.
Go to new share. Yeah. You can.
Yes.
Yeah. Thank you. Yeah. It's okay now.
Okay.
Yeah. So yeah, there are many applications. So we have some questions that I've already dropped here, even though they sent it to me personally. One is, what is the difference between bioinformatician, computational biologist, and a biotechnologist? So 3 things. So, and this is very clear because our people are still considering this new area, and maybe these clear definitions will guide our people to know what to focus on. Are they really want to be bioinformaticians, or would they want to be computational biologists, or would they want to be biotechnologists? And yeah, maybe if you could speak into this, it will guide.
Okay.
Yeah, thank you.
As I've said earlier in my presentation, bioinformatics and the computational biology are relatively synonymous, they are similar. But the thing is, bioinformatics basically deals with the genome and sequence. You know, we have the protein, as I told you, from genotype to phenotype. Okay, so bioinformatics is majorly what we call molecular bioinformatics, molecular biology informatics.
Okay.
So it has to do with the gene, the product from the gene, how they interact. Okay, whereas the computational biology is total, everything in which where computer is being used in biology. For example, I said earlier, maybe for example, when you use computer to do mapping of a particular area to know the kind of organisms that are there, or when you use computer to map, or when you when you design like all those epidemiological studies, where you use predictive model in statistics to predict. All this is still computational biology, but it's not bioinformatics. For example, let me give an example. There is maybe if I go by example, people will understand better. Okay, there's an outbreak of cholera in a particular part of Nigeria. Okay, then this cholera has been consistently occurring maybe for the past 10 years. So now, but there is data available. So now a computational biology in collaboration with statistics will, what they will do is they will get this data and then predict on the data using computational tools to predict, okay, maybe because of these factors, there will be this tomorrow. So those are computational biology. Those research does not involve the use of the DNA, does not involve the nucleotide sequence. And then biotechnology is actually a field where you apply bioinformatics. Okay.
Okay. Yeah. Go ahead.
Biotechnology is a field where you apply bioinformatics, because virtually every biotech research you involve bioinformatics. So bioinformatics is in biotechnology, but biotechnology is not inside bioinformatics. So it's just like a tool or a technique in which you use to achieve your aim in biotechnology.
Yeah. So, yeah, thank you, Dr. Akinola, for the explanation. Yeah, there is this, what I could call an intercept, okay, between the bioinformatician and computational biologist. And it's difficult to separate these. And maybe I will.
Okay. I think
Yeah, number one, it both makes use of computer science. Two, both makes use of biological data. So, but because bioinformaticians, we could say, focuses more on the DNA and the sequences, now computational biologists could also go into ligand studies, ligand studies and some other things. So we could say that computational biology is more broad, whereas bioinformatics maybe could be inside, but at this level, we could still welcome that computational biologists and the bioinformatics has a lot of intercepts, I mean, between the two and all of that. So thank you for that. The next question I could raise for myself, I think the question that the girl asked was also important to me, which you have responded to. Yeah, but I could see a lot of research questions that also came up in the course of your presentation, but maybe we could discuss that later. So the question here is, please, how does bioinformatics help in the area of animal husbandry? Okay, back to you, doctor. Animal husbandry.
It depends on your research objectives. Okay, animal husbandry. What do you want to do? Are you looking for the hybrid animal, or are you looking for animals that are resistant to a particular disease and pest, or do you want to boost productions? So it depends on what you're actually looking for. And in each of these areas, you can actually apply bioinformatics. Okay, how do you do that? For example, let's say you are looking for a particular resistant gene to a particular disease. Mm-hmm. Okay, you could actually, once you know the gene, there's a way you could, I think that's a genetically engineer your plant, your animal to make it resistant to that particular disease. Also, bioinformatics is being used in vaccinologies, where you can actually use bioinformatic tools to design vaccine, to actually administer these vaccines to your animal, or to your animals, to make them they are healthy, or resistant to a particular disease. So in the area, it could serve in a bio vaccinology. There's a few like that, which makes use of biotechnology. Bioinformatics. So it depends on your research question. Once you come with your question, then you'll be able to design what tools or what aspect of bioinformatics you actually use to do that.
Okay.
Does that answer the person's question?
Yeah, I think so, because there are a lot to do to improve. We could call it genetically modified system just to achieve high yield production. Even in some of the animals I know around here produce a lot of milk, but this is unnatural because already some things have been done in their genetic system, and they could give enough yield of milk. So some milk, particularly the one I saw in UK, is natural from the animal. And for animal to give you such raw milk that each time you go to the supermarket, this milk are there, it means that there's something extraordinary with the animal, which biotechnology has also played some role to make that to happen. Yeah. So someone also is asking, how does bioinformatics apply in the detection of multi-drug resistance in microorganisms? Okay, maybe we could take that as the last question. Then I don't know if Chiamaka Obas, if this is a question. Okay, how we could apply in plant breeding. I think you have responded to the plant breeding.
Yeah, plant breeding in the area of abiotic stress or biotic stress. Maybe if you want to know the particular gene that is resistant to a drought or to a pest, and then based on the Arabidosis, which is a model plant, you could study those those genes in them, and if with that effect, you could genetically modify your plant to that regards. Then in the area of AMR, which is a antimicrobial resistance, right?
Yeah.
Okay.
How can we do this? Yeah.
How can we apply? Yeah, bioinformatics, as I've said, bioinformatics is just like a tools you use to guide us to make reasonable maybe finding. Then you go back to the lab to validate it. So AMR genes are present in the microorganisms. So how do you find them? You have a particular microorganism that is not resistant. Okay, we have another one that is resistant. What you do, you sequence both their gene, the complete genome of both of them. Then you use bioinformatic tools to look for that difference, which gene that actually is responsible for that particular resistance, drug resistance. So once you identify the gene, then you go ahead to actually clone those genes, produce the gene, and look for an antibiotics to target the
so thank you. I think thanks for that. Yeah, before you be able to study the resistance, maybe it could also help to first of all take phenotypically if the organisms are resistant. Then if you confirm that they are resistant, then you can now go on to do PCR. So the steps involved is extract DNA, do PCR of the target gene, then sequence the gene. Then when you get your sequence, you could now run what we call a single nucleotide polymorphisms to identify the particular codon where the mutation has taken place and also the amino acid changes that have occurred there. So I think just know that it's very possible and it's very basic. I mean, to apply bioinformatics to do that, but that is also coming together of molecular biology and the computer. So first you start with molecular biology, then you end up with the computer to be able to say that this is resistant in terms of genotypic characteristics of these things. Finally, does a bioinformatician have to learn programming and data analytics, or is the job just to study already analyzed data? Doc, you still want to respond to this?
Yeah, bioinformatics, we are into, okay, maybe I didn't mention that. It's actually in two major areas. Well, I think I did. One, you produce tools for biologists to use. Secondly, you apply those tools. So now it depends on which area you want to focus in. For example, if you want to start developing tools for scientists to use to solve problems, then you need to learn some programming stuff, maybe like R or Python. Then if you want to be using already available application tools to solve a problem, just you apply them, then you don't need much programming. What you just need to do, just need to know the basics on how to run a Linux, how to write code on Linux. Then that you can actually to download those tools on your system and work with them.
Yeah.
So it's a two-way thing. It depends on your preference. If you want to start to produce tools for other people to use, like the XPath is a tool. So somebody wrote it, so to solve protein coding or whatever. So if you want to be doing that, then you need to have a strong knowledge of computer programming.
Yeah, so I've seen many people that are coming from mathematics and computer science and engineering, majoring in developing the tools, for us to use as biologists. So myself, I'm interested in first producing the data and secondly using the tools that already been developed by these guys to analyze my data. Yeah, I think that is very important. Maybe I could use this point to say in general, where do you start? If you have the case to go in this direction, where do you start as a Nigerian student and as a Nigerian researcher, as someone that is in Nigeria? Now, I needed to make this input because of the peculiarity of our environment and also how to thrive so that you will not be limited. The bioinformatics does not require much. It's laptop, internet, and also your thinking. So these are the three things that are involved. Then where to start, because you could see we are having some persons that ought to join the meeting couldn't join because of one, the internet. Two, the laptop may not be able to attract good internet that is already existing in the environment. So maybe I will say at this point that you might need to invest in good internet of good RAM. So basically, we could say the least RAM of your laptop should be 8 gig. Okay, it should be 8 gig. Then secondly, your phone, which sometimes could bring the network, should have good RAM of least 3 or 4 gig RAM. So these are the two things you need. Then you have the lab in your house. So Dr. Akinola has been on lockdown for some time and is working from his room, and it's not a problem. Yeah, because the
Yeah.
internet and everything is there. So I think you might need to invest in this. Sell anything you can sell, get these things for yourself. Then again, the internet, seek for 4G, where you could get 4G. Sometimes and some places, depending on the internet, you might not get access. Then again, is I've posted in the chat the link for the WhatsApp group of NGBC, NAU, Genomics and Bioinformatics Consortium. You could also join. A lot of training are going on. We make it public there. Then you can start from the basics. Okay, you can start from the basics and start learning. Then also, I will also say for those who have advanced and you are having metagenomic data or NGS data, so the NGBC also could assist in analyzing this data with you without pay, just to help you assist in using the basic tools that maybe you don't have now to analyze this. Okay. Then just to end also, I would also share a screen of one of the meetings where we want to apply bioinformatics. So, and is on molecular docking. So this is a meeting that will also introduce bioinformatics and begin to build those that are already in drug design. So this is also what we call the computational, because here we should be able to download plants, some ligands from plant database, chemical database, and some proteins also. So the molecular docking, it's all about the interaction of protein and the drug ligand, things like that, to determine the binding affinity. So every drug that you see and is doing well is because it has a good binding affinity, okay, with the protein and all of that. So this program will be able to guide us through. So, and we have a pharmacist that is in South Africa who has PhD in chemoinformatics that will lead in this workshop. It's a full workshop, not a webinar like this. And those who could go to Nnamdi Azikiwe University, the library will have a good internet, and you will see it. There will be teaching assistants that will be in the class. So it's both online and also on site, so that one could attend to your question. Now, of course, you can register to be a member of NAU Genomics. Then if you register, it means that for the 3 days or 4 days of this meeting, you will pay 2,000 naira. So it's not it's not so much. And we did it so because we are encouraging bioinformatics in the Southeast Nigeria. Then for those who are not members, yeah, of course, 46 people have already registered for this, but we can take more if you want to be part of this. So here we shall be applying bioinformatics to discover how you can also use bioinformatics, which we call in silico model, to develop, interaction between organisms, their protein and drug or plant-based drug, as the case may be. We have had publications using this tool in the past, but time will fail me to show this, but you can check the Google Scholar maybe you could see when Ebola came, the first response we did to Ebola was to look at some basic plants that we could use to tackle Ebola. And we had a good result. We also did drug repurposing. Today, ivermectin can be used also to attack some other diseases that initially it was not designed for. So it is this molecular docking approach that was used for this. So of course, the scope of the study is already outlined here. So you could look at it and start pushing in your career. So I think I can stop sharing now, and we have taken the last question, and to thank all of us that internet permitted to participate in today's webinar. We shall soon publish again because we intend to do this thing every month, once in a month. So next month, we shall have a bioinformatician from EMBL. Yeah, EMBL. He works in EMBL, who in UK, and also we have one in Germany here. So who will also take us in the next month. So we are still thinking about the topic he will take, probably available careers in bioinformatics, and also bioinformatics and the COVID-19, because bioinformatics have really done a lot in COVID-19 in detecting the variant. Now Nigeria has a peculiar variant because of bioinformatics. That is how we got to know. Yeah. So we'll see how it goes next month. But if you could join with the WhatsApp link that is pasted in the chat, then you could follow our activities and what we do to get ourselves knowledgeable in this area and this emerging area in medical sciences. Once again, I want to thank Dr. Akinola. Of course, the time variation between Nigeria and Canada is not funny. So he has to wake up in the midnight to really participate in this. Whereas we allow the Nigerians to sleep well and wake up in the morning. So thank you so much for your time. We shall knock at your door again. We shall knock at your door again in the NGS workshop coming by June or May. I don't know how the calendar say, but we shall check this up. Yeah, so that we can still, all our meetings now are on-site and online, depending on what is convenient for people. They can key in on either.
Okay.
Thank you so much for your time, and thank you for doing this for us. We are very grateful, and may God bless you.
So thank you too. Yeah. Thank you for having me.
Yeah, so thanks to all the participants today. Please, if you need the slide, and we recorded the whole thing, you can request using the NAU bioinformatics at unizik.edu.ng, and we shall send to you immediately the slides, and also so that you can review this again and again. So thank you, and see, I hope there's no other question. Okay, thank you everybody, and permit me to end the meeting now.
Okay.
Yeah, okay. Then please take note of the bioinformatics email address and send any other thing you will need across, and we shall respond to you. Thanks, and God bless all of you. Bye.
Bye.
Yeah.